Okay, welcome back. The last paper this afternoon is Steve Frank, local, on microbial pathogenesis and metabolism.
Thank you. In the first part of the talk, I'll describe several examples from bacteria and yeast, what we call microbes. And the point of these examples is that these microbial systems have essentially public good systems, in the sense that individuals in these populations will secrete molecules that are costly to themselves, but these secretions provide a benefit that's shared by everyone. So it has a classic public good structure. Individual costly, but public benefit shared by all. And consistent with the logic of public goods that everybody's familiar with, we do in fact see cheaters in these systems where individuals do not make secretions and are essentially cheating in the public good setting, but they still get to share in the public benefits. And so the question would be, with regard to evolutionary dynamics, what influences the frequency of these cheaters, these non-secretors? So I'll give a biological introduction where I'll show you several of these systems just to show you the kinds of things that happen in nature. And then the second part of the talk will be devoted to evolutionary dynamics, where we use a variety of classic analyses from evolutionary dynamics and evolutionary biology to understand how these systems evolve. And as I go along, I'll point out some of the relations between our evolutionary dynamics and evolutionary understanding, and some of the kinds of questions that arise in economics and in social science. And they're actually quite similar, very close. So again, the first part of the talk is just going to be background biology, and I'm going to show you quite a few examples, because if you're not a biologist, I think it's useful to see the kinds of things that actually happen in nature and draw the parallels to social systems to the extent that you want, but I'll talk about the biology. So the first example arises when bacteria or other microbes colonize a new habitat. For example, they infect your body, or they may also be colonizing a part of the environment, but especially you can think of infecting your body. And one of the problems for microbes to grow is that iron is often in very short supply, and the microbes need iron. So in the tissues in your body, in your bloodstream, and in your tissues, there's very little free iron. So this is a big problem for microbial growth. And so what many microbes do, for example, bacteria, well, first of all, this would be a host molecule that's called transferrin. So most of the iron in your body is bound to the molecules in your body. It's not free. And so for a bacteria to grow inside your body, it has to acquire that iron. And bacteria will then secrete molecules that are called siderophores. So an individual will secrete this molecule. This molecule will then diffuse away from the cell that secretes it, and it will float around in the environment. And this bacterial molecule, siderophore, can take the iron from, for example, transferrin, or pick it up free iron, and it will bind the iron. So now you have a molecule that the bacteria secreted that's bound to iron, a siderophore iron complex that's floating around outside of the cells. And that could be available for the bacteria and the microbes to take up. And so that's how they scavenge iron. A little diversion, because it's interesting, is that in your body, when you have these kinds of infections, you actually make a molecule that's called siderocalin. And this siderocalin can take the iron back from the siderophore iron complex before the bacteria get it. And it is a defensive molecule. It's a defensive approach to defending against infection. And, well, of course, the bacteria have to grow, and so they have ways around this. So, for example, bacteria such as anthrax makes an alternative siderophore that can avoid the siderocalin. So that's how anthrax grows. And the anthrax needs these alternative siderophores to cause a really severe infection. So this is the typical back and forth between hosts and parasites. From our point of view, though, the key issue is just the siderophore is effectively a public goods, right? The siderophore is secreted. The cost of secretion is borne by the individual that makes the secretion. Once the secretion is out there and diffuses away, it's available to all members of the population. So in the typical public goods setting, we would imagine there would be cheaters. And in fact, when you look at natural populations, there are cheaters, individual microbes that don't secrete. So that's our puzzle, is what affects the frequency of secretors and non-secretors. Those would be cooperators and cheaters if you wanted to use a game theory language, but this is just what happens in natural systems. So we're going to try to understand these problems. Let me continue on with the natural history background just to let you know more about the biology. This is an experiment done to demonstrate that the secretions actually have a consequence for the way in which bacteria grow inside the body of a host. And this is an experiment done in the laboratory where the experiment starts with a bunch of caterpillars that would develop into moths. But what happens is that at time 0, which isn't shown, each caterpillar is infected with some bacteria which have these secretion systems. They secrete the siderophores. And in the case where their secretion system is fully intact and all members of the population are secreting the bacterial population, then at the beginning of the experiment, all of the caterpillars are alive, 100%, and then the caterpillars die of the infection. And with the secretion system intact, they die very quickly. So this is, the quicker this drops, the faster they're dying, because it's the fraction alive. So this is the fully expressed secretion system. Now if you infect the host with bacteria that lack the secretion system, they die more slowly. So this is just empirical evidence to show that the secretion system has consequences for growth. And although this is kind of interesting, it's also interesting to reflect that when any pathogen infects your body, a virus, a bacteria, a protozoan, infectious diseases, essentially all of them have a large number of secretions that they make to manipulate your body at different stages of infection. When they first invade, they have secretions and mechanisms that are specific for invasion. As the infection develops, they manipulate your immune system. Many of these are by secretions that diffuse away from the individuals and act at a distance, so they're very much public goods. A large aspect of infectious disease has to do with these what are called mechanisms of pathogenesis, many of which are secretions of these forms that have this public good structure. So these are not isolated problems, kind of interesting that you can just pick out, but they're really fundamental to the biology of these organisms. And it's just in the last few years that people have begun to really think about the evolutionary dynamics of how these systems evolve and what the consequences are. So the first question then would be, that I raised was, why do individuals secrete these molecules if the cost is borne by the individual, but the benefit is shared equally by everyone? An individual that didn't secrete wouldn't pay the cost, but would get exactly the same benefit as everyone else, because they're diffusible molecules, not tied to the original secretor. If you look in the current literature, there's a very strong, clear, pretty much single answer to the question, which is that it's kin selection. And the idea to a biologist is very standard. It would be simply that within any particular population, the individuals are very closely related genetically, typically. And if they're closely related genetically, then think what would happen if an individual cheated, it didn't secrete. An individual that did not secrete would grow faster because it's not paying the cost of secretion, and it could outcompete its neighbor that's making the secretion. So it would gain a benefit, but it would be outcompeting a neighbor that's very genetically similar to itself. And in biology, the currency of success is transmission of genes to future generations. That's what causes the evolutionary change in various characteristics. So if an individual outcompetes a neighbor that's genetically identical to itself, that's not going to cause an evolutionary change. So there's no advantage. So the competition with closely related individuals you could think of as being between groups. A group that's more efficient is going to produce more progenitors, more descendants. And so really the competition is for efficiency rather than internal competition. By contrast, if individuals were not very closely related and they outcompeted a neighbor, then that would cause an increase in the frequency of the particular genes of the non-secretor. So the argument is that kin selection is a very powerful force. If you're an economist, it's correlated equilibrium. You can simply say, if individuals are genetically similar, they're going to have similar strategies. If they have similar strategies, then they're not going to be able to outcompete a neighbor because they're essentially doing the same thing. And so kin selection and correlated equilibrium in these settings are really identical conceptually and in terms of dynamics. So depending on your background, you can look at it either way. But they're effectively the same ideas. So does this work? Well, the answer is yes. We know that kin selection is a very powerful force in nature. If we run an experiment in the laboratory, and people have done this, here's a particular experiment. The experiment starts with 50% cooperators, those are secretors, 50% non-secretors, those are cheaters, if you want to call them. You start an experiment in the laboratory with 50% secretors and non-secretors, and then in every generation, you grow up the bacteria in a beaker, and you have a bunch of beakers. And in one experiment, in each beaker, you put individuals that are very closely related, that descended from a recent common ancestor. So you know that the genetic similarity is very high within each beaker, but there's genetic variation between the beakers because you start the beakers with different individuals. And you run that experiment and just let them evolve, and the secretors rise in frequency. That is to say, cooperation rises very quickly. It works. It just says that the theory of kin selection is a good, solid theory. We know that. And if you use the bacteria as a little calculator, they calculate the answer correctly, which is to cooperate. And if you mix them into beakers, then the relatedness is low and cooperation declines. Did you want to ask a question?
I can see that would work for zebras, but I thought bacteria mutate so quickly.
We'll get to that.
Oh, okay.
But you have to wait about 30 minutes.
Oh, zebras can't wait that long.
Okay. In fact, on the next slide. So this, if you look in the literature, there's a lot of experiments being done, a lot of discussion, and pretty much everybody who's working on this is kin selection is the issue. You can do if you isolate kin selection as a force in the laboratory, it will absolutely work. And we know that kin selection is a powerful force in nature. But it's much too narrow a view of social cohesion. So we know kin selection is an important force in social cohesion. But we also know that you can never understand kin selection outside of the context of what I like to call demography. Demography is how long colonies live, what are the flow of resources, and so on. If you're a biologist, this would be life history. If you saw Simon Levin's talk last week, you're going to see that this is discounting of future events. These are all classical biological problems. But it's interesting, in the history of the biological literature and cooperation, people know that kin selection and demography are both very important, but typically they get treated one or the other as if they're different fields. And I did a lot of work 15 years ago showing that any reasonable analysis of a real biological problem is always going to take into account both forces. Everybody knows that. When you look in the literature, it's not consistently applied. So one of the things I'm going to do, and I've been doing recently, is just consistently applying these concepts to these microbial problems. So there's no great new theory. These are standard theories in biology that are now pretty well understood. And then the next issue is mutation and timescale. So we'll get to all of these points. But I want to give you more biology, because if you're not a biologist, I think it's important to see really how important and widespread and deep these phenomena are, all the way down to anything in where evolutionary dynamics occurs. And I think you can see that by looking at microbial systems. So I'm just going to give you a few more examples. Biofilms, maybe you've heard that word, even if you're not a biologist. Let me tell you what they are. When bacteria colonize a new habitat, this could be a part of your body, or it could be the pipes in your house, because there are biofilms in the pipes in your house. They first start to grow, not all bacteria, but some bacteria. They first start to grow, and then eventually many species will start to secrete a little goo. It's a biofilm, what we call an extracellular matrix, technically. And so the bacteria grow up inside of this biofilm as an aggregation inside of this goo, and eventually then they'll start to disperse out. And this biofilm is interesting from a biomedical point of view, which is for the following reason. It was known for many years that certain chronic bacterial infections were very hard to cure with antibiotics, but it wasn't really understood entirely why. And it's really only in the last few years that people have now found that in many cases the bacteria are protected inside these biofilms, and it's very difficult to deliver antibiotics into the biofilm. So biomedically, this is a huge subject of great importance. But with regard to just natural history, it's also quite interesting. If you think about a pipe where water flushes through it occasionally, if the bacteria are not attached, the water flushes through and out the bacteria go. But if they're attached into a nice sticky biofilm, they're going to be able to stay in that pipe for a long time. The biofilms have an interesting consequence demographically. They increase survival. The bacteria won't be washed out. At least they increase the ability to stay in a habitat. But they probably often reduce reproduction because resources diffuse into these biofilms relatively slowly. They're all together very closely, and so resource flow within these biofilms is probably lower than it would be if the bacteria were out free in the environment. So to some extent, it's a trade-off of survival, increased survival, probably for reduction in reproductive rate. So it's very strong demographic consequences of these biofilms. But they're quite interesting, and also the secretions are made, they're public goods. An individual that secretes, the secretion's out, any other bacteria can get into the goo and use the benefits of the secretion, and there are cheaters. That is, individuals that don't secrete the biofilm. So this is another public goods problem.
And these biofilms can be multiple species, too.
Getting there. But yes, it's a very interesting question. Next slide after this one. One other interesting thing that was just discovered in the less than a year is that in a single species biofilm, in this particular case, grown in the laboratory, what these people found was that this is a biofilm of a single species of bacteria. They found that as the bacteria grew, that there was a signaling and differentiation process in the following sense. The bacteria first colonize, and they're not making biofilm. They grow, and they secrete a signal. And so that signal begins to build up in concentration. And when the signal builds up in concentration, then some of the cells secrete a surfactant. A surfactant is something that changes the surface properties, tension properties of a liquid. So surfactant gets secreted. The surfactant builds up in concentration, and the surfactant signals some of the other cells to secrete the biofilm, the extracellular matrix. The cells that secrete the extracellular matrix don't secrete surfactant, and the cells that secrete surfactant don't secrete the extracellular matrix. So we're getting specialization and differentiation. Now, so this is basically specialization of tasks happening autonomously among bacteria. And if you're a long-time evolutionary biologist, I think that's spectacular. Maybe it doesn't sound that spectacular, not a biologist, these things are things, but these are bacteria that are single-cell autonomous, and they're differentiating. One of the great problems in biology is, how did life start and go to something like us? Our cells are incredibly complicated. They have specialized tasks. We just think of our bodies having all these different tissues. But life didn't start that way. Life started, obviously, primarily more simple than bacteria, And this is actually the first example that I know of where we're getting the spontaneous emergence of this differentiation in signaling. And this is a huge part of modern biology, is to understand signaling networks and regulatory properties. And the fact that this is occurring in biofilms among autonomous cells is, first of all, spectacularly interesting from an evolutionary perspective about the conditions that would favor such emergence of specialization. Does this happen in all biofilms? We actually don't know. Does it happen in some biofilms and not in others? What are the conditions that allow for spontaneous emergence of differentiation and regulatory signaling among autonomous cells? Okay, so this is an opportunity to study what are some of the really great puzzles in biology in terms of evolutionary history, mapping onto a lot of what modern biology is, what some people call systems biology, which is regulatory signaling and differentiation, and systems that can be manipulated and studied and are tremendously diverse. Again, another diversion, but evolutionary biology is a very hard subject because everything that's interesting happened a long time ago. But here, the things that are interesting that we think of as happening a long time ago, maybe we can actually study them in real time with variation among different species. And so this is a real opportunity to get insight into some incredibly interesting evolutionary problems. This is really very new. This is just something that's just published, so this is a field that's just starting. We can see these things. Multiple species in biofilms. So this is a plaque on your teeth. Think of it as a biofilm. Plaque on your teeth form. First, what happens is that there are some bacterial species that specialize in binding to your tooth. So they get started. And then there are bacterial species that bind to the bacterial species that bind to your tooth. So you get a little ecological progression. And there are species that bind to the species that bind to the species that bind to your tooth. So we know that happens. That's cool. But it's more than cool because I can say for sure, but we don't actually know the details, that there's a lot going on in there. It's not just all a happy little aggregation. There are species that are having positive synergistic effects with other species in what we call mutualism loosely, although that's a very misleading, oversimplified term. There's going to be competitive effects where one species is killing off another species by kicking out toxins so it can take over its habitat. Within each species, there's going to be individuals that are secreting and not secreting and cheating. Kin selection is going to be going on and all the other forces of social cohesion. So this is just has, in microcosm, almost everything that's interesting about ecology and evolutionary biology is happening in these biofilms. And it's of tremendous medical importance, so there's a lot of study being done, but it captures almost everything that we're interested in evolutionary dynamics, and many of the problems that are in the economic and social sphere have at least analogies. You can draw the analogy as tightly or as loosely as you want there. That's somewhat of a personal preference, but certainly you'll see many of the key forces involved. Another thing that's now well known is what's called quorum sensing. So quorum sensing is if a bacteria is going to colonize a habitat, and initially there's only a few bacteria, if they start secreting siderophores to get iron or biofilm, it's not going to be of any value because the number of siderophores that are secreted by a few cells, they're going to be so small they're just going to diffuse away, and they'll never come back to any of the cells in the population. It's just concentration's too low. So when they're in low density, there's no advantage of secretion. And so the way they handle this is that first they secrete a very cheap signaling molecule. And when the population builds up and the blue signaling molecule builds up in concentration, then that triggers the secretion of the costly siderophores, biofilms, and other effectors. Okay? And then remember that in infectious diseases, in the history of an infectious disease in your body, secretions are being turned on and off at different stages of infection. So this is one particular kind of density sensing, but there must be many sensoring systems that turn on and off different secretions that have different dynamics in the history of an infection. This is, again, a huge field. Understanding infection at the molecular level means understanding the mechanisms of pathogenesis, and these are at the heart of the kinds of things that are happening in pathogenesis. Now, you might imagine that with a signaling system like this, there could be cheating, and there's two levels of cheating here. You could not secrete the initial signal, so that's called signal negative, or you could not secrete in response to the signal, and that's called signal blind, because you don't see the signal and respond to it. So there's two levels of cheaters. Both things occur in natural infections, and people have studied in the laboratory. And here's just some data that are kind of interesting. A study done in the laboratory on mice where they start bacterial infections in the skin with a quorum sensing system. Signal negative, there's two different experiments here. In the first experiment, in the infection that started in the laboratory, almost all the bacteria put into the infection have a full quorum sensing system, but a few individuals are signal negative, so they're not making the initial signal. Over the course of a week, the signal negative types are increasing in frequency to 15%. That's consistent with the notion that the secretions are costly. So the individuals that aren't secreting are outcompeting their neighbors and increasing in frequency. Now, presumably the reason why not every individual in the population is a non-secretor is because the infections that have a large number of non-secretors are going to grow more slowly, and they'll have a lower probability of starting a new infection. So you have these sort of two levels of process. Now with the signal blind individuals, same experiment, but they increased to about 35%, which is consistent with the notion that not making the secondary secretion is associated with a higher cost or savings. So the two-stage system of cheap signals early, expensive secretions later. So that's consistent with the notion. Again, then what's the question? If you look in the literature, people say, well, we can understand why quorum sensing occurs. It's kin selection. Same experiment done on this, but a different system. Again, if you run these experiments in the laboratory, they work. You start with 50% quorum sensing, full system, 50% that have cheating of one kind or another in the quorum sensing system. You run these out, each time step is a generation. You run them in beakers. High level of relatedness, cooperation evolves to a high level. Low level of relatedness, cooperation declines. Again, the bacteria, you're using them as an evolutionary calculator, and it works. Kin selection works.
What does high and low mean?
High relatedness?
Oh, I mean, roughly numbers?
Oh, well, relatedness wasn't measured, but basically you start with a recent common ancestor, and so the amount of genetic variation would primarily be from mutation happening within the beaker. But you start with basically a very small number of cells, or you mix a bunch of cells from different beakers. In other words, a beaker grows up, and then you take a very tiny number of cells and you move
I
into a new
understand the
beaker.
mechanics. I mean, it's kind of curious that even at the lower relatedness, it's not declining very fast.
Yeah.
That was true of the last.
Well,
one.
that could be, I don't know that, yeah, that obviously stands out, and we would have to go back and look at it. But it could be the case that in the absence of quorum sensing, that the fitness is so extremely low. So it has a lot to do with the marginal shape of the costs and benefits, probably. It could also be that low relatedness is not anywhere near zero. It could be still fairly high. It depends how they did the experiment. There's only two treatments here. They don't measure relatedness. They just give you a design. So this is the last key problem I'm going to talk about in terms of natural history. It's what I'm going to use for the rest of the talk. And this is something that I find quite spectacular when I first read about it, and yet in some ways it's so obvious. And this is the trade-off between rate and yield. And the idea goes back, the idea has to do with metabolism. Metabolism is how your body or your cells take up resources like sugar and break that sugar down and make it into energy. And so the way we do that and the way bacteria and yeast do that is about pretty much the same. The fundamentals of metabolism evolved a billion or more years ago, and we pretty much have the same structures in terms of basic breaking down of resources into energy. And what's happened in the last 10 years, people have begun to think about this a little bit, and somebody realized and had really built up this discussion that with regard to basic metabolism, there's this fundamental trade-off of rate and yield. So imagine that we take some bacteria or yeast and we throw them into a vat of sugar. And so the cells, each cell is going to suck up some sugar across its, what we call cell membrane, so the cell's like a little sphere. It's going to suck up the sugar from outside into the inside of the cell. It's going to break that sugar down, make energy, and it's going to use that energy to run its business, which is basically growth. But now think about driving that reaction faster. Sucking up the sugar faster, breaking it down, making energy faster would be a good thing, right? The faster you go, the better. But it takes energy to go faster. If you're going to go faster, you've got to pull the resource across the cell membrane, and you can do that. You have to make more transporters, and there are transporters, literally, on the surface of these cells. And you might have to use more energy to pump the sugar across the cell membrane. And if you're going to drive your reactions faster, it takes more energy, because that's just basic chemistry. You can drive them faster, but you're going to have to put energy into it to do it. So you can drive the reaction faster, but the more energy you put into driving the reaction faster, the less net energy you're going to have at the end. So there's fundamentally a trade-off between rate and yield. Yield we would measure as, for every gram of sugar, how much new gram of biomass can the cells make. So there's a fundamental trade-off between rate and yield. And this fundamental trade-off between rate and yield is interesting in a variety of perspectives, all of which I find quite fascinating. If you think about the fundamentals of metabolism as being just sort of the building blocks of life, and people have been trying to understand metabolism for a long time. When I was trained in economics and social biology perspectives in biology, and of course I also learned about metabolism. And it never occurred to me to connect those two things in my mind, even though I was sort of trained to think all the way down that life has these conflicts and cooperation. But metabolism I always had on the side. That's just chemistry. And yet this rate versus yield trade-off has implicit a very standard social trade-off. It's just the tragedy of the commons. If you're competing with a neighbor, you're going to suck up resources faster to outcompete your neighbor in that vat. So if I'm a strain and I suck up resources faster than you do, then I'm going to outcompete you, even if my efficiency is lower. So that's just a tragedy of the commons. It's just like saying there's a common field of grass. It's not owned by anybody. I can put out a bunch of animals, and my rate of sucking up energy off of that grassland is the number of animals I put out. So I can increase my rate by putting out more animals. If you want to compete with me, you're going to have to put out more animals, and pretty soon that grassland the yield is zero. We know that that's a common tragedy. That is the tragedy of the commons, literally, because it's the common field, the commons. And so this is the trade-off is fundamentally the tragedy of the commons. So in that sense, there's a social perspective, and I'll talk a bit more about that. And then in another sense, it's interesting because, as I mentioned, modern systems biology, much of modern biology is looking at the regulation of metabolism and what goes on inside of cells, all the different reactions, all the proteins involved, how they interact, and so on. And there's two schools of thought. So people who are doing this, they're measuring lots of things, and it's like, okay, it's very complicated. And so even biologists now recognize we're going to need some math, we're going to need some models to understand these things. That's what systems biology is. And so modeling metabolism, there's sort of, to be crude, there's sort of two schools of thought. You can model, if you have to make a model of metabolism, you can make the assumption, a priori, that the flux of metabolites through the system, that's just the rate at which things pump through the system, is maximized. That's called rate maximization. If you make that assumption, you can work out a whole bunch of models and predictions about the structure of metabolism. That's fine. There's a whole school of thought like that. Then there's a school of thought that says, well, maybe yield is maximized. So there's a whole other modeling school of thought that looks at the design of metabolism from a yield maximization perspective. But they're just a priori assumptions that people make because one person says, well, rate, another person says yield. But in fact, we can see that there's going to be a trade-off, and that natural selection is going to push the design of metabolism along that trade-off for all sorts of reasons. So again, to me, this is just, it's so simple, and yet it's kind of spectacular because it's the heart of life, contains all of these issues. And yet it touches not just social aspects, but it touches really almost all of modern biology in terms of the questions that people are asking. So making these connections I've just found really interesting as I started to read this literature. So this is an example of a yeast that grows in some sugar. This is the rate of sugar uptake. This is the yield in grams of biomass made per gram of sugar taken up. The yeast are grown under different conditions. And without going into the biological details, because there's some interesting details, but if you just grow these yeast under different conditions, they fall along the rate versus yield trade-off. So it's a real trade-off. It's not something that's just an artificial construct, but it's empirically something you can measure. That's a single species grown under different conditions. Now, people have gone out, not a lot, but there's some studies, and collected different yeast species that are really quite different. They live in different habitats, they're quite different in their physiology. And they've grown them up under common conditions in the laboratory. And if you grow them up under common conditions, some of them are fast, they suck up the resources fast and they have low yield. Some of them are slow and they have higher yield. So the natural history is pushing this very basic metabolic system of sucking up sugar and turning it into energy and yield along that rate yield trade-off, it appears. We don't really know a lot about what's going on here, but you can imagine that different conditions might favor rate or yield. So, what are the conditions? So that's what I'll talk about.
Do these species compete?
Well, some of them may. I don't actually know the natural history. Right? We have a yeast guy here.
I don't know. I only focus on one dot.
I might conclude from what you were saying originally that something like rate times yield should be maximized, or the output at the end. But then it seems to me that maybe these are more like R selected and K selected.
That would be a closer
type.
first pass argument.
And that therefore this distribution is the result of some competitive displacement.
It's not necessarily so, though. You can get along that rate yield trade-off without competition between the species. And I'll show you.
I mean, but otherwise, why wouldn't everything just sort of converge to whatever's the best point?
Because they could have different demographic conditions. And I'll show you that demography has a huge effect on the rate yield trade-off, for example. And that's what I think is going on. Rather than kin selection, I think it's demographic primarily. Let's see. So, no one has done this experiment, but I'm sure it would work. If you run these organisms in a beaker in the laboratory and you increase the relatedness in just the kind of experiments we looked at, you would select for higher yield, because that's just like a tragedy of the commons. Correlated equilibrium, high relatedness, whatever you want to call it, tends to favor a cooperative outcome. Cooperation, roughly speaking, is yield maximization, because rate is primarily a competition against neighbors.
But it's yield per what? Yield
Per gram of sugar, for example, biomass per gram of sugar.
Well, is there a time unit associated with it, per generation or something?
Yeah, you could do it that way. Yield per gram of sugar per hour.
I'm just wondering, because if you increase the rate, doesn't that factor into the overall yield, or because you get more generations per unit of time or something of that sort?
Right, it's net yield for any fixed period of time. We can go back. I don't know what the units are. Gram of biomass per gram of glucose. It's,
Per hour.
well, yeah, it would have to be. So anyway, people have talked about that, but nobody's done it, but I'm sure the experiment would work. But in general, again, that's isolating the factor of kin selection outside of the notions of demography and other evolutionary factors that we know are very important. And so that's what I'm going to do for the rest of the talk, is discuss some of how these various forces would play out in order to give us some sense of what might be going on. But again, nobody's done these experiments, so we don't really know, but this would give us a framework in which to understand these issues. So I wanted to look at this rate versus yield trade-off as my example, because I think it captures a lot of the notions that we're thinking about, but it could also apply to quorum sensing or siderophore secretions. Concepts are pretty much the same, but let's make a model that is sort of loosely based on the rate versus yield trade-off. And so I wanted to make a specific model, and so here it is. In this model, there's a concentration of resource, which is a sugar X, and the sugar is going to be taken up by the microbes at a rate Y. Y is the amount of transporter that's present. So of course, it takes more than transporter to take up resource, but just for simplification, we're going to take Y as the concentration of transporter. And we know that in competition, microbes do increase their transporters. Okay, so the whole regulatory system for transport's upregulated, so this is quite realistic in that regard, although the model is still very simplified. So we're going to imagine that the concentration of the transporter is going to be proportional to the rate of uptake. So the rate of uptake of the resource is proportional to Y, and a fraction F of the resource is going to go to making more transporter, which is going to increase the rate of uptake. And a fraction 1 minus F of the resource is going to go to making biomass. So that's the trade-off between rate and yield. So we're just looking at how an organism would split the resource into rate and yield in terms of making more transporter are making more biomass. It's a model, but it's not too far off from fundamentally the issues that cells have to deal with. Now we want to understand the evolution of F, the trade-off, so we'll set up a competition because we want to understand how this is going to occur under biological competition and evolutionary dynamics. So there'll be alternative types that would have a trade-off F prime, and that would lead to different levels of transporter and biomass. So that's roughly the setup, and this sort of maps onto a set of differential equations. I'm not showing you any of that. I'm just going to give you a rough idea of how things go. In order to understand and calculate an optimal value, we need a measure of payoff, and in biology, the classic measure of payoff is fitness. But what's fitness? Well, the standard measure of fitness is given by this equation here, which you can think of as the summation of survival times F is fecundity or reproduction. So the idea is, if we want to figure out what the consequence of a particular change in physiology is for fitness, we have to say, how does that change in physiology affect survival, say, to next week, times the amount of reproduction next week, plus the probability of surviving 2 weeks into the future, times reproduction 2 weeks into the future. And so if you saw Simon's talk last week, he talked about that in terms of discounting. Discounting is a standard property of evolutionary analysis. And so this is just a standard measure of fitness, which accounts for the discounting issues. People know that, but they don't always take it into account when they study biological problems in a very direct way. If you have a constant death rate, then the discount rate or survival rate is e to the minus dt probability. And we're going to take reproduction as proportional to the amount of biomass. And so we just have a simple measure of fitness to apply in this model. Of course, you can do it in different ways, but from our point of view, all that matters is that there's an appropriate discounting of the future. And we'll see how that matters here, for example. So these are dynamics of the growth. So you imagine in this first case, we have some sugar, a fixed amount of sugar. We throw some microbes into the sugar, and they start to grow until the sugar's gone. Here there's no competition. There's two curves, but each case they're grown alone. So they're not competing with each other. So they start to grow up when you put them in the sugar. And in this case, if F equals 0.04, then crudely speaking, that means 40% of the resource is going to making more transporter, which increases rate of uptake, and 60% of the resource is going to biomass. So if they do that, they grow up relatively slowly, but to a higher ultimate yield. If the amount of resource that's put into increasing rate is higher, then they grow faster, but they grow to a lower yield because they're putting more of their energy into making transporter rather than biomass. So we can see the first demographic consequence very clearly. If colonies survive a long time, these are yeast growing in different habitats, and the habitats are ephemeral, but some habitats last a little longer for some species, and some habitats last not so long, for example. If the habitat lasts a long time, then high yield is going to be favored over high rate, and this is a simple example. I'm not taking into account all factors, just looking at this one factor of growth dynamics. On the other hand, if the colonies don't survive very long, then high rate is going to be favored over high yield. This is our pure clones. The kin selection coefficient is 1. The correlation between strategies is perfect. This is just purely demographic discounting of the probability of death into the future. So this means that demography by itself is potentially a sufficient explanation for all the variation in rate versus yield. It's probably not the only explanation, but it just shows as the extreme opposite of the claim that kin selection explains everything. My own personal view is that in terms of natural history, demography varies tremendously. And so we're going to get a tremendous amount of variation in rate versus yield. Remember, this is the fundamental design of the basic units of metabolism on which really all of life is based, is going to fluctuate powerfully based on the different demographic properties of the population. Now, if we throw those two strains together in competition, the high rate type outcompetes the high yield type very easily because the high rate type sucks up the sugar. By the time the high yield type starts getting going, all the sugar's gone. Okay. In this case, there's just a fixed amount of sugar. And you could vary that, but basically, high rate outcompetes high yield on direct competition. So we can see that there still is a competitive game aspect to this. And we know that when we grow in the laboratory, microbes under competitive circumstances, they evolve to upregulate their transport systems. And people know the genes that are involved, the regulatory mechanisms, and so on. So this is a repeatable phenomenon that's quite reasonably well understood molecularly. So it happens. So if we take that model I showed you, and we figure out what the optimal value of the rate versus yield trade-off is, taking into account the expected survival time of a colony, that's the demographic component, and the expected level of kin selection. So this is the level of relatedness. You can think of that as the correlation coefficient in the strategies or behaviors of the types, or the correlation in the different genotypes. It's literally just a correlation. Okay? If the correlation is perfect, then this is the curve that maximizes fitness purely demographically, under different demographic circumstances with no competition. As we start to add some competition in by some genetic mixing, we get a rapid increase in the tendency to favor rate over yield. So that's a tragedy of the commons sort of thing, right? You're getting a decline in yield strictly in response to competition, because this is maximizing yield down here, or fitness, ultimate productivity for the group.
What are those labels, 1.0, 0.9?
It's the correlation coefficient between the different strategies in a correlated equilibrium setting. You can think of correlated gain. It's the correlation of the strategies, pairwise interactions. It's not, roughly, that's a good first approximation from a game theory point of view. So you can see the powerful effect of kin selection, but you can also see the powerful effect of demography. So they're both important, and that's a normal outcome for any sort of model like this. So, but you can see that kin selection coefficient of a half, microbes probably have high relatedness often, but a half is the equivalent of full siblings. So that's still a high level of relatedness. So that says that any reasonable amount of genetic mixing is going to completely break down cooperation in these sorts of systems. So that's sort of the interaction between kin selection and demography. Let's look at time scale now in mutation. This gets to your question. I told you about it, half hour. So now the experiment's a little bit different, the setup. We're going to have a continuous flow of resources into an environment that would be like our bodies. When a microbe gets into your body, there's continual renewal of resources. We're going to throw in a clone, no genetic variation, just a clone. We're going to let it grow. So it grows up and increases its biomass over time. But the clone is occasionally going to make some mutants, say at a rate of 10 to the minus 5, that's 1 in 100,000. Cell divisions, there's going to be a mutant that's going to be produced. And some of those mutants will have a different trade-off of rate versus yield. They will maybe have upregulated their transporters, so they're going to suck resource in more quickly. So eventually, there's going to be a mutant that sucks up the resource more quickly, and it's going to outcompete the original progenitor. So that means that if the colony goes long enough, it's inevitable that the descendants are going to outcompete the original progenitor. So every clone and colony is going to face competition from its own descendants. And I think that's, and so eventually there'll be that competition. So in this case, the key parameter is actually not the mutation rate, but it's this number up here, which is the rate of cell death. So in this model, particular example, cells last about 1,000 time steps before they die. So that's only about 4 turnovers in the population. And that's important because, well, let me show you what happens. If we increase the rate of cell turnover by a factor of 10, so the average cell's lasting 100 time steps rather than 1,000, there's more cell turnover, and you'll see that things happen faster even though the mutation rate's the same. And that's because the key parameter is not the mutation rate, but it's how fast the cells die and then can be replaced for the mutant clones to take over. Because what happens is these things grow up so there's almost no sugar. Even though the sugar's flowing in, they're sucking the sugar up at equilibrium so fast that the amount of free resource is very low. So the growth rate is very low and things are very limited, which is a typical property of these sorts of growth dynamics. So the cell turnover, which is a demographic property, is really the key factor, not the mutation rate. You can think of this in terms of relatedness if you want. You can say that the relatedness is breaking down by mutation, and that's an accurate description. But I prefer to think of it as individuals essentially are going to be competing with their own descendants. And we know that that's the case in multicellular organisms. Our bodies have about 10 trillion cells, and over the course of our lifetime, we face potential competition with our cells growing faster and outcompeting the other cells in our body. We call that cancer. And a huge amount of the cellular physiology in your body are checks and balances to protect you against cancer. So it may be very well that you're paying quite a large cost to protect yourselves against the descendant cells in your body. And this is something you can see where this comes from. Now, that I've thought about a lot in cancer, but it never occurred to me to think of microbes as actually potentially facing competition from their descendants, and maybe a certain amount of the design of the microbial genome, and I have no idea, I don't know that anybody's even talked about this before, that the design of the microbial genome has to do with dealing with the fact that a clone will eventually compete with its own descendants. It's just like a multicellular aggregation very similar to cancer. It's really an analogous problem. People have talked about the role of protecting against mutations and robustness, but this is different. This is competition against descendants. So to the extent to which this is important in microbial design of genomes, I don't know, but I really don't even know that people have thought about it, as far as I can tell. So if you do a model, you put all this together, we've got expected survival time, optimal trade-off. These are clones now, there's no relatedness factor. They're clones starting in a batch. And we look at how long they survive. Here the turnover is 1 in 1,000 steps, so that's about 4 turnovers. This is the mutation rate. This is the optimal value of the rate versus yield trade-off. And the longer things go, the more the initial progenitor is favored to raise its rate and decrease its yield. It's raising its rate, which is basically a reduction in productivity, in order to be able to compete with its own descendants. It's a cost that's imposed by the mutational competition with its own descendants. There's no other advantage to increasing rate in that way. That's just like our bodies, the extent to which we pay costs to regulate our tissues to protect against cancer is essentially a cost. If we didn't have to do that, we would have a lot more, perhaps, energy or other resources to deal with to grow faster. So if you compete an individual that didn't have to deal with cancer protection versus individuals that do, all other things being equal, presumably the individual that didn't have to regulate against cancer would grow faster. So now that's a turnover of 1 in 1,000. Remember I said that the key parameter is the rate of turnover, a demographic property. If we increase the rate of turnover by a factor of 10, you're seeing now that already the rate versus yield trade-off is becoming dominated by competition against descendants of the progenitors. So you see the potential power of the force involved here, very much shaped by demography in this context. So this is the timescale conflict coupled with the demographic properties. Again, in some sense, this is all very standard evolutionary dynamics, and yet it's just not consistently applied to these microbial problems, which are so attractive because they have all of these factors in them. So I see this coupling together of all the biology I talked about in the first part with these simple concepts as being really a very interesting field going forward. There's a lot of people working on this, and so this is, by no means have invented this, but I do think that the standard theory has not just been applied as in the simple way that I'm showing you. So these are these 3 factors, and I'm going to take 5 more minutes and give you one more factor. Some people are fading, but most people are still awake. Quorum sensing. Remember, the key in quorum sensing, and this is very important with regard to cooperation, is that, a little bit like Rob's talk, it's conditional behavior. It's not that there are cooperators and non-cooperators, it's that they're adjusting their secretions in response to the conditions. It's a conditional behavior. And as a modeling issue, Rob's point is actually a very important one. It happens over and over again in biology where people forget that behaviors are often almost always conditional responses to the environment. They're not hard-coded behaviors, even in bacteria. And so you get a lot of modeling controversy where somebody makes a model with hard-coded behaviors and then they argue with somebody else who's actually captured the biology fairly accurately and they argue forever about it. But it's important to understand that because it's really critical, and we'll show the importance of the difference of having adjustable behaviors here. So quorum sensing is a very particular kind of adjustable behavior. It says, don't make the secretion until the density builds up. But there are many adjustments. That's just one kind of adjustment. Remember I told you that in infectious diseases, these pathogens are turning on and off secretion, some in invasion. There's actually a few that are sort of reverse quorum sensing. They secrete in low density during the initial invasion because there are certain molecules that are important for getting the infection started, and then they turn it off as the infection builds. So it can go either way. So there's many of these conditional properties. And so I wanted to understand this because it's very important. And so here's a model. It's a bit different from the previous model, so we're sort of starting over now. I wanted to make a model of the growth of a colony and understand the role of cooperative behavior at different time periods in colony growth. And so I used a classic setup, which also if you saw Simon's talk, you'll recognize. It's a very classic setup. You can think of it as an example. Think of a colony of social insects. So you have a queen, she mates, and she's going to start a new colony. She has no workers. So the first thing she does is make some workers. And those workers then get together, they get some resources, and they make more workers. They don't make reproductives, they don't reproduce. And so you go through a period of colony growth where the colony grows very fast, workers make more workers, and the colony grows as fast as it can. And then eventually when the colony gets large, reproduction begins. And so that's, Simon mentioned, that's the classic bang-bang strategy of control theory. You invest in one strategy of growth. Once you get to a certain size, you switch your strategy entirely to reproduction. Very common in biology. And so let's look at that because infections go through this property. Many colony growth patterns of insects growth, of microbes go through this. Now, let's suppose that survival is proportional to the colony size, which would make sense. In small social insect colonies, they're in trouble. They don't survive very well, so they need to grow rapidly because survival is very low for small colonies. Small individuals is often the case, their survival isn't good. So if we imagine a behavior at one point in time, just a simple incremental behavior that's expressed once at one point in time and then turned off, if it's expressed early and it increases colony growth, then the colony size will be higher for all future times, even though the behavior is expressed once. Right? So survival carries forward through time. That's typical. If you get bigger, you'll be better off all through future periods of time. If you invest that energy as a one-shot reproductive effect, though, reproduction is usually means putting energy into something that's going to go away, and so it disperses out. So the one-time reproduction doesn't carry forward to future benefits for you demographically. So there's a very big distinction between survival and reproduction in terms of the consequences for long-term fitness and success. And so we can think of that in terms of marginal valuation. Early in the colony growth cycle, the marginal gain for survival is very high because that behavior will increase your colony size at that time and through all future times, because once you're bigger, you'll be bigger through your whole growth cycle. Whereas an instantaneous increase in fecundity gives you some benefit, but that benefit is dispersed out. So you're getting the fact that survival benefits tend to decline as the organism grows larger for investment in survival, whereas fecundity benefits stay relatively steady. And that's what gives you that bang-bang strategy. But it's interesting now to think about in terms of something like a microbial secretion, where how are we going to understand how conditional behaviors are going to change through the course of the colony life cycle. So let's just make the assumption that a 1% increase in colony size causes a theta, which is a parameter, times 1% increase in colony survival. So we can study how changing size changes survival and how that changes investment in cooperative behaviors. So we can think of a cooperative behavior that can be done early in colony growth, or a cooperative behavior that can be done later in colony growth, and look at the selective pressures on cooperation early or late in the demographic cycle. And here's just what would be the solution, running out a simple model, so I'm not showing you the details. This is theta equals 1. That means 1% increase in size, 1% increase in survival. This is the level of relatedness, so that's the correlation coefficient. So a half is like full siblings. 0.05 is like hunter-gatherer groups, so that would be typical perhaps of a human society. And traditionally people would say, well, a relatedness of 0.05, something like in simple human societies, is too low to explain cooperation by kin selection, which is true. But look at what happens if we look at the level of cooperation. You can think of this as secreting a siderophore, of a quorum sensing system, any sort of cooperative behavior that's a public goods behavior. This is just a generic type of cooperation. Early in the colony growth cycle, because growing a little faster causes such a benefit that carries forward through all time steps, the level of cooperation is quite high early in the colony cycle. But it declines later in the cycle. Now that's pretty easy to understand intuitively. If we got thrown out of a boat in a storm, and we were all thinking we were going to go down, we would hop to in terms of doing the various duties on the boat to make that boat get through that storm. We wouldn't be worrying about who's getting a better place, who's going to get a few more resources, because we're all going to go down. So cooperation is pretty easy when everybody's in the same boat, right? And yet, as the storm abates, and then that's the end of the cooperative bout, then conflict might emerge because there might be limited resources, limited amounts of food, for example, on the boat to survive. So it's actually quite a standard sort of situation that might emerge. And it could be very typical of biological problems where early in a colony cycle, there's a tremendous synergism in survival for working together and doing cooperative behaviors, and that declines. So you see the tremendous amount of cooperation that can happen. Now this, just 1% increase in survival for 1% increase in colony size. If we increase that to 2% survival for 1% increase in colony size, we're seeing the cooperation go up. And by the time you're up to 3, a relatedness that's as low as a hunter-gatherer society where the relatedness is really pretty low, we're getting early in a colony growth cycle a huge amount of cooperation between individuals. That's a demographic consequence of the natural fact that colonies, groups go through life cycles as a group. So relatedness is very important, but it's always in synergistic effect with demography. You can never extract the two. Again, it's a basic, obvious point, and yet there's a lack of consistency in the application. People talk about it, Simon talked about it, but if you look in the literature on microbes,
which have tremendous variation in demography. You don't see the consistent application that people run this kin selection thing out, because kin selection, social behavior kind of go together as a slogan almost. But in terms of basic fundamental principles of dynamics, you can see these things go together. So those are the forces then: kin selection, demography, mutation, and time scale, what I call adjustable competitiveness. That was the last thing. So it's not a long list, but it's long enough that it gives us a pretty rich understanding of evolutionary process. It applies to microbes, and I think in ways that will be very informative in the coming years. And I think it's also a pretty good framework for a lot of the public goods problems that people in economics and social science are interested in. So just to review, because I talked about a lot of things, I started out telling you about siderophores and the way in which bacteria scavenge for iron. This is a public goods problem. I talked about biofilms. Remember, this is of great biomedical importance, but the biofilms have tremendous demographic consequences for changes in survival and reproduction. So there's going to be, and they have this properties of differentiation. So this is really getting at a lot of interesting issues of spontaneous organization, of specialized roles. This is the ecological situation where we're having multiple species coming together. We really don't know all the pushing and shoving that's going on in there, but you can be sure that there's a lot of it within and between species. Quorum sensing is a very important issue. That's a student in my class wrote up a little note saying that recent studies have shown that as much as 5 to 10% of the genomes of microbes are regulated in a quorum sensing way. So I mean, this is a huge amount of the genomes of these microbes are being adjusted in response to local density. You start to think about it, and I started to think about it, and so these people in my class have really taught me a lot. Decomposition. You want to decompose a leaf? Well, a microbe can't suck up a leaf, so they secrete enzymes. Once you secrete an enzyme, it's a public good. So a lot of fundamental decomposition is probably the sort of public goods situations that might be related to these quorum sensing systems. If you're all alone and you secrete that enzyme, you're probably not going to be able to digest enough, depending on the diffusion properties of the enzymes, if it's a liquid or you can do it locally. So a lot of scale effects. A lot of interesting questions. So anyway, people in the literature, I've said this many times, say kin selection is the answer, and it's a factor, but I showed you in this simple model, which has a survival and fecundity components, that demography is very important, discounting, if you want to say it that way. Kin selection is very important, but they interact. Then we looked at the timescale conflict, where individuals are competing with their own descendants, and that's having tremendous effects on the rate versus yield trade-off. And really a completely open question as to the extent that the design of genomes are affected by this sort of phenomenon. And then ultimately the role of colony life cycle in the degree of cooperation that goes on. And so I think these things together really provide us with a lot of opportunities to study these things empirically in microbes and perhaps in other kinds of questions as well. Thanks. I will, if I can find my water.
So if you're going to have mutation and individuals are going to compete with their offspring, I guess the benefit function, the discounting ought to have a relatedness, which also decays with time.
Yeah, some people like to think of it that way. They like to think of it as a decaying relatedness. And there's no question that you can do that. That's a reasonable way to do it. I think that it's, to some extent, when you actually do it, you have to kind of force it to back calculate the relatedness, whereas it's pretty straightforward just to look at it as a dynamical process over time with mutation and competition. But you could, without question, unravel it and reformulate it in terms of the decay and relatedness. So that is an accurate description. So it is a double discounting, if you want to say it that way.
Are any of these quorum sensing genes on plasmids, and would that make a difference in how to think about this?
I don't know the answer. Certainly I can imagine the selective pressures on plasmids being somewhat different, because the plasmids will often have different interests from the host cell that they live in, and so there could be a conflict in terms of between the main chromosome and the plasmid chromosome.
Okay, but especially with the multi-species plas, that there might be even stronger selection for quorum sensing because they're being exchanged between species.
Mm-hmm. And it's interesting, again from my class, somebody wrote up a little note, something I hadn't thought about, but the siderophores, once a microbe secretes a siderophore, any other microbial species can take it up, potentially. So there seems to be a lot of polymorphism in the structure of the siderophores and the uptake of the siderophores. And in fact, somebody wrote up a little note in their class, again, just spectacularly interesting, that some insects seem to use the scavenging of the siderophores of pathogens. So you have a bacteria that infects you, and you have other bacterial species that live in you as symbionts, as sort of mutualistic symbionts. And your mutualistic symbionts suck up the secreted siderophores of the invading pathogen, and that protects you from infection. Biology's great.
So I was wondering, thinking about the commons problem as engendered by the great deal problem. So think about Rob Boyd's talk now. Could we imagine evolving punishment strategies for overly speedy consumers?
Yeah, people have thought a lot about, there's just a paper, it just came out where people did that, but I mean, the question of, I tend to differentiate between punishment and what I call repression of competition. So I think that, and they're different. So repression of competition, for example, could be a randomization mechanism. So like meiosis, if you know what I mean by that, right? That's a randomization mechanism. That represses competition in the John Rawls sense that all entities are behind the veil of ignorance, and therefore they can only increase their success by increasing group efficiency. I don't consider that punishment, and yet it has the same powerful effect of increasing group cohesion. So there's punishment and there's repression of competition. Now the question in all biological systems, and Rob's talk really brought this out, isn't that these things could work. The question is, how are the mechanisms structured so that they're effective and they won't break down? And so it's all it's really all in the biological structure and the mechanism. So randomization is one of the few real success stories of repression of competition in terms of forming group cohesion. And we know a few examples of it from nature, but not necessarily a lot. So, but it's an open question, and it's a topic that's really taken off in the biological literature over the last 5 years. You can find a huge number of people discussing it. I've long wondered whether the slime molds, I don't want to go into too much, but the slime molds are the ones that they get together in an aggregation of cells, and they differentiate between cells that make a stalk, non-reproductives, and cells that reproduce in the spores. And I've long wondered whether there's a period of randomization just before they differentiate. Now that would be a randomization, but not a punishment form of bringing the interests of individuals together in that regard.
Yeah. Well, I suppose you could imagine just that the animal is programmed to do, there's a program of one thing you do if you happen to be in certain environmental circumstances, and another thing you do in others. And if those two things are closely paired, you would look for, does the pair of strategies succeed well? But they'd have to stick together. They have to be on the same chromosomes.
Yeah. Well, it depends whether they're conditionally expressed or whether they're genetically expressed. In which case
Well, I would suggest because
a lot of
the genetics.
Yeah, a lot of the microbes are, the amount of recombination in microbes varies from almost none to some. So there are microbes that have very little recombination. So there's the dynamics of genetic models are quite different from the classical sexual models that you might learn in standard population genetics.
As a political scientist, I just find quite fascinating the last comments, of course, the notion that nature has read Rawls even before Rawls wrote it is just really interesting. And of course, when Rawls talks about it, he talks about behind the veil of ignorance as justifying principles of justice.
Yes.
Here the argument would seem to be that behind the veil of ignorance essentially leads to efficiencies. That is to say, if you don't know whether you're a bottom swarm slime mold or top slime mold in advance, then you basically work for the good of the slime molds because you don't know whether you're going to be on the top or the bottom. I think that's a really, really quite neat, a neat point, and maybe in fact has relevance to the political science, political theory literature, because it suggests a quite different reading of justifications for behind the veil of ignorance, rather than thinking of it in terms of principles of justice, thinking in terms of principles of efficiency.
So the first person that I know,
like Sunny rather than Rawls.
Yeah.
the first person that I know who wrote about that extensively was Richard Alexander, and then Brian Skirge wrote a book that deals with this extensively. So there's some literature, and, everybody
No,
comes through.
But the randomization point you just made, at least for me, wasn't.
Oh, definitely,
That's a new one to me.
Yeah, no, Alexander talks about it a lot,
Mhm.
and he's the first person in my mind who really actually had a clear notion, even in biology, of the tremendous scope and importance of repression of competition or punishment, sort of the whole scale of things. How if you reduce the opportunity for conflict within groups, it's an obvious point, right? If you reduce the opportunity for conflict within groups, the only way for individuals to increase their success is you said it.
Mhm.
And there's lots of ways, there's many ways it can occur. People understood randomization. Egbert Lee was the first person, I think, who was quite explicit about it. But it was only known for a few rare biological examples. And Alexander was the first person to draw large-scale analogies, both to humans and across all different biological systems, at least in my mind. And so now this notion of repression, competition, punishment is at the center of a lot of studies in biology, not just the way Rob talked about it in humans, applying evolutionary principles, but throughout all organisms. The question now is, to what extent do these systems explain group cohesion in simple organisms, at what level, in social insects, and microbes, and so this is a really open question. And it all sort of has exploded out from there. Actually, I have a review I wrote about 6 years ago. It's the biology, but it reviews the history of the biological part of it in relation to, actually, Adam Smith has something that's not far from this. So, it's a famous quote that people use all the time.
So randomization itself becomes a public good, which could conceivably.
No, but it is, absolutely.
Could be selected for if they're bioturbation mechanisms or something, and you might, just like in Rob's, you might be selecting for randomization.
Yeah, I mean, I like my classes who are biologists, because if they're biologists, they're trained in genetics before they get to me as a graduate student. And so they all know genetics, and they all know that in genetics you get a gene for your mother and a gene for your father, and they nicely separate, and that's just how genetics is, right? But it couldn't have been like that to start with, right? It was probably a free-for-all scramble, because that's how life starts. And so sometime in the history of life emerged this incredibly orderly, the phrase that biologists use to teach it, even though they don't mean anything about roles or anything about it, they call it pheromiosis. That's the phrase in the genetics books, pheromiosis. And that was done by people who have no interest whatsoever in social sciences or any of those sorts of things. They'd be horrified by the notion that it's not just a biological phenomenon. But it was always called pheromiosis because it is a randomness, because it was recognized that it was a split in a fair sense. And it's amazing because when you get taught it, as that's just genetics, like that's just not things work. But if you actually begin to look, there's cheaters. We call it meiotic drive. Those are chromosomes that outcompete, so they get into all the babies, and the partner from the other parent gets into none of them. And one of my favorite experiments, we should probably let people go, but I'll finish my experiment. One of my favorite experiments is somebody took one of these meiotic drive chromosomes. So it's like an X and a Y chromosome. So the father has an X and a Y, and it makes a gamete, which is going to make the baby. If it's a Y, then the baby's a boy. If it's an X, the baby's a girl. It's a fruit fly. And so it took the Y chromosome, and it took from a different chromosome, which is a different part of the genome, something that made the Y chromosome get into all the babies, and the X chromosome get into none of them. So these guys are having all sons. So you put them into a cage, and you grow up with flies, and pretty soon there's no girls. They're all boys. Well, this cage isn't doing very well because they're not reproducing because nobody's making eggs. And some of the cages went extinct. But in a lot of the cages, different genes across the whole genome evolved to repress the driving Y chromosome. Because any gene that repressed the driving Y chromosome would then have a sex ratio that's 50-50, and those daughters would be the progenitors of a large fraction of the next generation because there's almost no other daughters there. So because of the frequent expensive sex ratio, that shows in a very simple, direct way that in a short evolutionary period of time, the genome restructured to repress that competition. And that standard pheromiosis, which is taught as that's just genetics, is actually the outcome of intense conflict and competition with an entire genome, the genes spread all over the genome, that are involved in imposing that pheromiosis. Some groups have a lot of examples where that pheromiosis breaks down, and in humans it doesn't seem to. So in mammals it doesn't seem to happen very much. We don't understand why, but we will probably in 10 or 20 years with genomics. So yeah, there's a lot of interesting things, but that's, I'll get your question later. That's probably enough.
Actually, the bus is coming in a while. Bus is coming in about 10 minutes.
you want to stop or not?
Whatever it is for the people to
Thanks
know.
very much again.
Yeah, so you better let them go.